Hello everyone, my name is Viktor Дрёмин. I am a postdoctoral research fellow in the University of Oulu in Finland, and I am going to talk how we use Stokes vector polarimetry for screening of breast samples with different types of tumor. But at the beginning, I would like to tell you a little bit of information about our city and our university. Oulu is located on the coast of the Gulf of Bothnia. As you can see, Oulu is situated in the north of Finland, just over 550 kilometers from Helsinki and close to the Arctic Circle. Oulu is a unique place. In summer, you can watch the sun around the clock, and the aurora is also not uncommon for us. And of course, you should visit Oulu as soon as the difficult situation in the world ends. University of Oulu is one of the biggest and the most multidisciplinary universities in Finland. Our university was founded in 1958. Research and educational community consists of 16,000 students and 3,000 of university staff. Our optoelectronics and measurements techniques unit is specialized in demanding electronic applications. The research focus is on biphotonics and optical measurements. An actively developing area is printable electronics. The variety of applications has led to cooperation not only with other units within the university, but also on a global scale. And here you can see our large campus. This is the building where our research group is located. Okay, let's get to the scientific part. The outline of my talk includes these next sections. A brief introduction that includes statistics in breast cancer, about the developed experimental system, and a bit of polarimetric theory. Next, the experimental results will be presented, including statistical analysis. I will tell you about the first results with ex vivo measurements of human breast cancer samples. Last decade, there is a strong increase in the number of patients with malignant tumors, and breast cancer is the leading cause of cancer-related death in women, with 40,000 deaths a year related to breast cancer. However, millions of women are surviving and thriving with breast cancer when found and treated early. Such conventional imaging techniques like computer tomography, MRI, PET, they show very promising results for diagnostics of cancer. However, currently the only way and gold standard to set definite diagnostics of cancer is histology. Typically, the biopsy is taken at the time of surgery operation with a high demand of immediate qualitative pathological review. Otherwise, the repeated surgical interventions is required and leads to a worsening of the prognosis of the disease. Development of an efficient diagnostic modality may revolutionize current practice of cancer diagnosis and treatment. The ability to recognize the suspect tissue in a short time without interruption of the operation is able to increase the effectiveness of the surgery of the cancerous tumors. The standard histology technique is very laborious, very time-consuming and expensive. It takes very long preparation of the sample. First, it's cut into small pieces, then it's fixated, dehydrated, embedded in paraffin, then it's sectioned using microtome, it's mounted on a glass plate, it's rehydrated and stained, and only after that the observation takes place. So the whole process takes few days or weeks, and accuracy strongly depends on the skills of pathologist, and it only provides qualitative information. We suggest using the properties of polarized light for the analysis of cancer samples. This will not only reduce the stages and time of analysis, but also provide quantitative information about the sample. We have developed an experimental system for scanning cancer samples. The measurement system consists of illumination and detection channels. The light from the supercontinuum laser was used in the illumination channels. The probing wavelength was selected with the acousto-optic tunable filter, providing the possibility for the selection within the range of 450-650 nanometers. The linearly polarized light from the output of the filter was altered by a quarter wave plate into the state of right-hand circle polarization and focused with the lens onto the sample at 55 degrees angle. We use unequal angles of incidence and detection to eliminate spectral reflection. The diffusely reflected light from the sample was collected by the measurement channel at 30 degrees angle at a variable distance between two channels with the 20 times objective lens and analyzed by the Stokes polarimeter system. Automated spatial scanning of the samples was performed with the XY translational stage. Here you can see overall view of the experimental setup in the lab. Due to a high sensitivity of polarized light to scattering variations, the polarized spectroscopy has a great potential to be used for screening malformation changes in the biological tissue samples. Circular polarized light was used due to its unique ability to preserve the polarization properties during larger number of scattering events than the linearly polarized light. In this study, we propose measurements of the Stokes vector and the calculation of the degree of polarization as a diagnostic criterion for the separation of healthy and cancer tissue. Stokes vector consists of 4 parameters. The first one is the intensity of light. The second one shows how much there is horizontally polarized light over vertically polarized light. The third one shows how much there is diagonally polarized light over anti-diagonally. And the fourth one is the difference between right circle polarization and left circle polarization. Using the Poincare sphere makes it easier to visualize multiple polarization states at once for comparison purpose. Each point on the sphere represents a unique polarization state. North pole corresponds to the right circle polarization, equator to the linear polarization, south pole to the left circle polarization. The intermediate state, elliptically polarized light. Degree of polarization shows how much of light is polarized within the whole light that is detected. Okay, um, 15 formalin fixed and paraffin embedded breast cancer samples with their histology were provided by the All University Hospital Biobank. They included 13 samples of fat tissue, 11 benign fibrosis, and 5 samples of intraductal cancer or ductal cancer, carcinoma in situ, and invasive cancer or invasive ductal carcinoma. This slide shows the measurements of early stage breast cancer samples or intraductal cancer. This slide demonstrates photo of paraffin embedded sample, histological image of invasive ductal carcinoma, and spatial distribution of the normalized reflected intensity, the normalized Stokes vector, and degree of polarization of the detected light. Here we used a 450 nanometer wavelength in the illumination channel. As you can see, the most contrasting and informative image is the degree of polarization. However, the remaining measurement parameters, Stokes vector, can be used to display data on the Poincare sphere. The red dots are highlighted area in the DOP image. Next, we studied the effect of the source detector separation on the value of the DOP parameter. By the variation of the source detector separation, we could go from single to multiple scattering. But what is the optimal distance between the source and detector for our setup? We came to the conclusion that the greatest differences between different parts of the sample can be achieved with a separation of 100 micrometers. Here we used several probing wavelengths You can see the increased contrast when probing 450 nanometers. Thus, we used this wavelength to measure all the remaining samples. Next, you can see scanning of developed cancer samples or invasive ductal carcinoma. This slide also demonstrates photo, histological image of invasive ductal carcinoma, and special distribution of the DOP of the detected light. There is a nice difference between different areas of the sample. Also, you can see color-coded Stokes vector on the detected light on the Poincare sphere and expanded area of the whole data cloud. This slide shows Stokes vector corresponding to cancer region, fibrosis, and fat tissue. You can see the difference in the position of the points in the sphere space. Partially polarized light, which can be considered a superposition of polarized and unpolarized light, is represented by a point within the volume of the Poincare sphere. We have shown that circularly polarized light scattered within the breast sample is sensitive to the presence of cancer cells and provides the information close to obtain it from the standard histological examination. However, no staining and no slicing is required in our case. We are currently working on improving the resolution of our system to meet the microscopic examination standard. This will allow to distinguish the influence of individual cells and their nuclei on the polarization parameters. And finally, a statistically significant difference exists between four types of tissue in the DOP parameter. The observed differences may be related to an increase in the average size of the nuclei in a more developed type of cancer. This leads to higher anisotropy and an increase in successive forward scattering events, which reduces the degree of polarization. Also, the use of linear discriminant analysis showed high sensitivity and specificity of the method. Thus, a circular polarization imaging system for obtaining histological images was developed. Circularly polarized light scattered within breast samples is sensitive to the presence of cancer cells. The highest perceptual contrast between cancerous and normal regions was observed for the probing wavelength of 450 nanometers. The DOP of the reflected light was found to be the most sensitive parameter indicating the malignant changes in the considered samples. The average DOP values for intact fat tissue were 95%, for intraductal carcinoma 70–80%. The presence of invasive carcinoma reduced the DOP values to the level of 60–70%. Mapping the Stokes vector of backscattered light on a Poincare sphere helps to highlight the changes of polarization state, and the proposed methodology demonstrates a great potential for developing a new diagnostic protocol for routine clinical practice. It's our acknowledgements, and thank you for your attention.